Hi, I'm Dr. Anna Pfaff, and I'm an analytical scientist on the medical device extractables and leachable teams at EAG Labs in St. Louis. And today, I'm going to tell you how to turn on easy mode for the identification stage of your next E&L study. Our team performs chemical characterization of medical devices according to ISO 10993 Part 18 guidelines. We identify chemicals extracted from a device that might end up in a patient's body and the potential worst case quantities the patient might be exposed to while they use the device. These identifications are essential for a toxicological risk assessment later on, which is a major step on the way to getting the device approved. I specialize in liquid chromatography, high-resolution mass spectrometry, high-res LCMS, which is an extremely powerful analytical technique that yields a wealth of information I can use to pinpoint which compounds are present in your test article extract and at what concentration. Using this technique, I can determine the chemical formula and structures of each compound extracted from your device, along with an understanding of how compounds behave under certain experimental conditions. This detailed structural information enables me to identify each species in the extract as a specific compound that can be examined during a TRA. For example, if I were to analyze eye drop solution, I might observe species with a chemical formula that consists of repeating units of 2 carbon atoms, 4 hydrogen atoms, and 1 oxygen atom, C2H4O. If this makes sense in the context of the other data from our E&L study, I could identify the species as polyethylene glycol, which is a likely component of the eye drop solution itself, as polyethylene glycol forms a water-soluble gel that's useful as a lubricating agent. My job is to identify every compound above the analytical evaluation threshold, or AET, in the device extract, especially those that might be harmful. These compound classes are called cohorts of concern, COC, and they too have distinctive formulas or features that we can pick up with high-res LCMS. If a chemical formula has, for example, a high nitrogen to oxygen ratio or a high degree of unsaturation, we need to be on the lookout for azo or nitro compounds, which are potential cohorts of concern. And in the absence of additional data on the test article, we will undertake a very meticulous investigation to determine whether these cohorts of concern are present or not. So not all identifications are straightforward. Those of us who identify compounds for E&L projects know that each project has a difficulty level, a lot like our favorite games. So let me explain. For easy mode projects, we're able to efficiently and accurately identify compounds in an extract. And although we strive to provide quality results as quickly as possible for every project, easy mode projects are a guaranteed win, and they almost always go out on time, if not ahead of schedule. However, we often end up playing on hard mode when we lack sufficient information to confidently identify the majority of species within the agreed upon timeframe. Unfortunately, we don't have access to the difficulty settings in the identification game, but you do. There are some key pieces of information you can provide to make sure the team identifying compounds for your next project are playing on easy mode. To illustrate this, we'll use a real-life example to understand what differentiates an easy mode project from a hard mode project. But first, we're going to see what identification on hard mode looks like. Below is a real screenshot of the data table reporting some of the most abundant species in the test article. A lot of it's redacted for confidentiality. Each row represents a separate identification that I need to figure out. I show 8 rows here, but there are over 100 more. And for a handful of compounds in this project, the preponderance of data supports the identification as a species of low toxicological concern, such as a common surfactant. But for approximately 80% of the compounds in this project, the chemical formulas are consistent with potential cohorts of concern. For these compounds, there will be one of 2 outcomes. I will either need to, 1, determine that the compound in question belongs to another non-COC class of chemicals, or 2, determine the exact chemical identity of the cohort of concern and precisely how much is in the device extract. In either case, this additional investigation and review needed to arrive at either of these conclusions for each potential cohort of concern multiplies the time and effort required for this stage of the project from hours to days or even weeks. This would be a textbook example of a hard mode project. So how can you avoid this and turn on easy mode for your next project? Well, just open up settings and enable communication with the EAG medical device team. It's actually almost that easy, and here's why. Successful identification scientists are always trying to understand extracted compounds in the context of the test article as a whole. We try to relate the compounds we observe in the test article to rational sources based on the materials of construction and processing steps. ISO 10993 Part 18 guidelines emphasize the utility of information on the materials of construction and that it can and should be used to identify compounds in the extracts and to determine the likelihood of cohorts of concern being present. Providing EAG labs with information on how your device is made can turn a hard mode project into an easy mode project. So what kind of information do we need? It's anything that answers the following questions. So what are the components of the device made of? Are they nylon, PTFE, or silicone, or something else? How do you know they're pure? Quality control documents or certificates of analysis can shed light on sources of impurities or contamination. How are the raw materials processed? What chemicals are used during these processes? Examples include initiators, antioxidants, surfactants, lubricants, or other additives that might be left over from the manufacturing process. And finally, how is the device packaged, stored, or shipped? A device may be pristine before it's in the package, but species such as slip agents can and do transfer by the time the device makes it to our lab. So now that you know how to turn on easy mode, let's see how it can make a big difference for the outcome of the identification game. These slides reference the same project as the previous slides, but now we'll watch what happens when the client is very responsive and transparent. Here, they've provided several examples of data that is useful in identification, including identities of raw materials, synthetic processes, and reaction schema. And with this information, I was able to tease apart complicated chemical structures into building blocks that match perfectly with the test article raw materials. And with tools like ChemSketch and Compound Discover, I could sift through thousands of spectra and chromatographic data points to extract exact mass, fragmentation patterns, and retention time. And I could turn the project into my own personal sandbox to figure out which of these blocks were present in each observed species, in what order and in what abundance. So this level of communication with our client helped turn this table into a table that looks like this, where the potential cohorts of concern were instead determined to be consistent with the various collections of building blocks that make up our materials of construction. And by the end, we had 5 confirmed, meaning the compound happened to be part of our reference standard mix, 119 confident, and only 2 tentative IDs for this project. Identifying this many compounds by LCMS was still lots of work, but it was a fraction of what it could have been without close partnership with our client. And as much as we scientists like a challenge, delivering quality results on a tight schedule is our ultimate goal, and making identifications on easy mode helps us do just that. Thank you.